looking at phytochrome and this idea that when they were doing this experiment with lettuce, they were able to manipulate when it would germinate. Lettuce needs light to germinate. When sunlight is striking the plant body, there's slightly more red light than far red light. And so here you can see that the red light was allowing the lettuce to germinate, whereas the far red light, if that was the last quality of light that the plant saw, so to speak, it would not stimulate germination. And so you could keep playing with that over and over, manipulating red, far red, but it doesn't really matter. What matters is whatever quality of light the plant last was exposed to, that's what's going to cause the reaction of germination or not. So if you look on all of these images, it's always the far red that is keeping them from not germinating, whereas the red is allowing those seeds to germinate. And so simple experiments like that were what kind of led to this idea that there must be a pigment that is assessing that ratio of red to far red. And they did find it. It was phytochrome pigments, and it is kind of unusual because it kind of toggles back and forth into different forms, physical forms. And so the so-called phytochrome sub R, the P sub R, that's the form that is capable of absorbing red light. And then when it does it, and when it does absorb that red light, it switches physically into the so-called phytochrome FR form. So the P sub FR form, so that looks physically different. And when the sunlight energy is striking the plant, depending on if it's getting full sunlight or maybe it's getting shaded out a little bit, the ratio of red to far red light striking the plant body will be picked up by these phytochrome pigments, and then they assess the ratio of their pigments. They're able to determine what they should do based on that information, that ratio. And so we looked at this last time, I think, and we were looking at, this is the visual of what we were just talking about. If you take the phytochrome form of the pigment and strike it with red light, let it absorb that, it physically changes to the P sub FR form. And then again, hitting the P sub FR, now that form of phytochrome is optimally shaped and designed to absorb far red light. And then that toggles it back to the P sub R form of the pigment. And so this idea is that when they studied it a little bit more closely, it seems that it's the P sub FR form that's the so-called active form. What they mean by that is that it's the form that will go to the nucleus and start interacting with proteins, transcription factor proteins that work with the DNA and alter gene expression. So you can see here, they're showing nicely that it's a physical difference. There's this shape shifting going on between the more like the M shape for the P sub R form, and when it absorbs red light, it shifts more to a Y shape. And then that's the form of the pigment that will go into the nucleus. So this was an unusual group of pigments, but it really does help the plant assess what's going on with sunlight around it. And it is actually a way for them to tell how long they're in the dark versus how long they're in the light. And that can be really important so that plants, like especially around here, plants can assess seasons. It's a way for them to, there's other ways too, but this is one way that they keep track of, summer, spring, fall, winter. They can assess light versus dark, because as you know, it's getting darker earlier and earlier now with sunset in the fall here. And so they're able to assess the time of the year essentially with the phytochrome pigments. And that's super important for plants because that will tell them when they should go ahead and start the flowering process. And so you don't want to make a bunch of flowers when winter is right around the corner. And so you have to have a way to assess the appropriate timing. And probably just from out hiking around that some plants, they flower in the early spring, and other plants, they flower more toward the end of summer. And again, that is phytochrome pigments, that particular species is one of the ways that they're assessing when they should go ahead and flower. And when we think about forested ecosystems, of course, there's all kinds of complexity to the quality of sunlight these different plants will get, depending on if they're a very tall tree and they're way up where they're the tallest in the whole ecosystem and they're getting, their canopy is getting full on sunlight. And then you think about some shorter trees or some shrubs and they're not getting full sunlight, they're getting shaded out. And so those leaves have to assess what's going on with that and behave appropriately to that signal. So if you think about, here's just a mock-up trying to show that, you know, the highest canopy plants get 100% sunlight on their leaves, and then you can see a progression to much less as you go down into the lower canopies. And so that is just a reality of plants that live in a mixed ecosystem like that. Here you're looking at this blue line is what would be the experience of the tallest plants in terms of all the qualities of light striking their body. They get the full on sunlight. And then you have to imagine if you're the tallest plant in the canopy, you know, there's certain qualities of light that you prefer for photosynthesis, right? Do you remember what two qualities they like for photosynthesis? What does the chlorophyll absorb? What colors? Red and blue. Yes, red and blue. And so this idea that if you're the tallest and you're getting all the qualities of light striking you, and you want to do photosynthesis, you're going to preferentially absorb red and blue. That means for the plants lower, they're not getting as much red light because the upper canopy is kind of hogging it, right? So they're hogging the red light, and so they'll let more of the far red light through. And that's what we see happening with plants that are lower down in the canopy. They get more far red striking their body than they do red, because the taller ones hogged all the, not all the red, but a lot of the red got hogged by the tallest plants. And so they're kind of showing here, this green line is the far red idea. So the lower canopy plants are getting that. The red is what the tallest plants are getting. So if the tallest plants are getting plenty of red light, that's going to shift their phytochrome to the FR form and will be the so-called biological response. Lower plants in the canopy, if they're absorbing more of the far red light, that means most of their phytochrome pigments will be in the P sub R form. And that's actually a signal to them. They, by that ratio, they're like, huh, I have more P sub R than I do P sub FR. I must be getting shaded out. And so if you're a plant that's getting shaded out, that could be really important information depending on what species you are. So some plants need highlight to really thrive. And so if you're one of those plants and you're in a lower canopy of the forest, and you're like, I'm getting a lot of P sub R in my body over the P sub FR, I'm getting shaded out, I need highlight, so I better start growing up or growing laterally to reach maybe the edge of the shaded area. And so plants like that will use that ratio to shift their gene expression to start growing. Typically, they'll start to invest more energy in growing taller, trying to get unshaded. On the other hand, if you're a shrub in the lower canopy and you're designed to be a low light plant, if you have a bunch of P sub R in your body, you'd say, okay, yep, I'm getting shaded, but that's okay because I'm adapted to shade. So it's not a big deal. All right, so it really does depend on the plant species, what this ratio will tell them to do. It's not like it always tells them to grow or tells them to do a certain thing. It just depends on the context of the situation. What kinds of species are we thinking about and their light needs? Are they highlight needed plants or can they handle shade? So it gives them a lot of information and allows them to behave appropriately. This table is just showing some of the other ideas that they've looked at for what the phytochrome can help the plant deal with. And it can be the germination idea, getting the seeds germinated or not, like with the lettuce example, or telling the plant to shift its body form to, when they say de-etiolation, that means stop growing so tall and instead, slow down your shoot growth and start investing energy in, leaves and branches. So there's a variety of different things here that they're starting to figure out what phytochromes do for the plant. And it definitely, do they have, that's interesting. It's not on here. Add into your notes here that one of the big things that it does is allow for the plant to determine when it should flower. So the time of flowering is really another big aspect of this. And in fact, I'm kind of surprised it's not on there. They must have it in a different table, more with the flowering information in the book. But basically, the plant is really paying attention to the ratio of phytochromes and using that to determine when it should go ahead and start the flowering process. Okay, so phytochromes are really an interesting group. One of the former students here, Jeremiah, that did his botany degree here, went on to his PhD at the University of Madison, and he actually was on the team of scientists who published in the journal Science on the structure of the phytochrome. So his part of his PhD thesis was looking at the three-dimensional structure of phytochrome. So he did a lot of good work with that. Okay, so another type of photoreceptor. So phytochromes they're capable of absorbing red and far red. Now we're looking at blue. All right, so blue light photoreceptors. And so here, there's one group called the cryptochromes, and we actually have these too. It's not unique to plants. Bacteria, mice, humans, they all have cryptochromes. For plants though, what it helps to do is slow down stem growth. And sometimes that's very appropriate. Sometimes you don't want to keep investing energy in your stem. And so cryptochromes help with that. They know that the cryptochromes are important in the 24-hour cycle that the plant experiences, that circadian rhythm. And so they're looking at that a little bit more. They know that certain pigments like anthocyanin, for them to mature fully, the cryptochromes are involved in that process. And they know that the cryptochromes are kind of linked with phytochromes. And so they will kind of have a team effort on certain plant responses. So here is the idea, again, with this image that they're showing. Oh, one more idea, solar tracking. So the idea of them being able to track the sun moving across the sky from sunrise, high noon, going on to sunset. The cryptochromes are important for that because the plant likes to move and face its leaves to the sun at a 90-degree angle to the incoming sunlight. And so having a way to track that over time really helps maximize the amount of energy, sunlight energy striking the leaves. Again, this visual is showing that it's a shape-shifting mechanism again. You have the blue light striking the cryptochromes and it causes a change of shape in the physical form of the cryptochrome. And then you can see here, there's a so-called active form that, again, the job of the active form of the phytochrome is to go ahead and move into the nucleus and interact with proteins that are controlling gene expression. Here, they're just showing the idea of stem elongation. So they're talking about hypocaudal. That's why I put this image here. If you're wondering what a hypocotyl is, it's the portion of the stem that's below the cotyledons. So that's what they call the hypocotyl. So here they turned on the blue light and you can actually see that it's stopping the shoot growth in that region. And so they're able to, they do have data, scientific data that's showing that the blue light quality is what's controlling that. They also know that there's another group of pigments that are called phototropins that absorb blue light. So it's not just the cryptochromes, there's also phototropins. And they've been heavily studied because they help control guard cell opening, which is huge for a plant. If you don't have good control on your guard cells, you're not going to be a very effective plant. So they know that the phototropins are really integral into that. I'll show you an image to demonstrate that. And leaf movements, again, that's really important because the plant wants to present its leaves 90 degrees to the incoming sunlight all day long. And so its leaf blade has to be shifting in response to that. Also, if it's getting shaded out, they can show that the phototropins help with that and can detect that and allows for leaf movements away from the shade. They call it shade avoidance behavior, I guess. And even they've noticed chloroplasts move around in cells, in the leaf cells. They move around in response to phototropins, and I'll show you some pictures of that. And then the seedling phototropism, I showed you a picture the other day where that seedling was bending toward blue light that was coming at it from one direction. And so that's what's going on when you put your plants in the window and then you don't move them at all. And after a while, you start to see their stems bending toward the light coming through your window. It's actually the blue quality of light that plant is paying attention to. And through that, it's able to determine which direction the light is coming from and start bending it so it can present its leaves 90 degrees to that incoming light. And it also interacts with this idea that you can get hormone shifting going on in the plant where it causes auxin. You know that auxin's involved at the synthesized in the shoot apex at the tip of the shoot. And it actually causes auxin, instead of flowing evenly down the stem, it causes auxin to shift physically over to the shaded side of the stem. I'll talk more about that when we look at auxin hormones in more detail. So they're finding out that the phototropins have some very important signals that the plant has to pick up on and respond appropriately. And again, it's just this simple shift in physical shape that's allowing for the response or the signal, I guess, to be heard. And so once it absorbs that blue light, it changes its physical shape. And then in this case, they are showing a phosphate event. So there's a phosphate group that gets attached. You see the P's here. The phosphates get attached, and that's a very common strategy. It's a strategy your body uses as well as plants to have this in bacteria. This transfer of phosphates is a way to get a signal to be heard and allow the plant to respond appropriately. So here is another idea of the blue light. B, they were not getting exposed to the blue light. And then with figure A, they were getting unidirectional blue light. And that's what's causing, what actually happens then is auxin's being produced in the shoot tip, and it actually starts to get shifted to the dark side of the stem, and it tells those cells grow. And so if you have the dark side growing, then you get this asymmetrical growth pattern where the dark side is outgrowing the lit side, and that's what's causing the actual bending of the stem. And you can reverse that if you go ahead and take away that unidirectional light and give them more uniform light, then they can grow back straight up. And so it's a way to, in certain times, change their growth, but then they can change back. It's not like a permanent thing necessarily. This is the idea of chloroplast movement within a cell. And so they see with low light, the chloroplasts tend to distribute like that, and they think that allows for better capture of light energy when they're in this position within the leaf cells. Highlight, though, if you really hit them with like extreme light, that can be damaging. Then you actually see chloroplasts hide from that highlight, so to speak. So there's the ones that take the brunt of it, but then all the other ones are kind of shaded out and hidden away a little bit more. And so highlight causes them to shift. And then darkness, they just kind of sink, so to speak, to the bottom of the cell due to gravity impacts. Looks like they're sleeping. So phototropins can do a lot for the plant. And here, what they did is they were measuring stomatal opening and what qualities of light, and it matches up with the blue quality of light quite nicely, where you get a big absorption peak. And so it matches up with the stomatal opening. And so, again, you know, if you're a plant and you're hanging out and sunrise happens and the sun starts striking your body, if you're a typical C3 plant, you want to open your stomata right away. And so here is the more complicated description of what's really going on. I'm not going to go into this in detail, but a couple of things to point out. So here is the blue light coming in. So sunrise happens, the sun starts striking the body of the plant. That's the phototropin protein photoreceptor. And then you're seeing phosphate transfer going on here, phosphorylation events. And you can see this is showing nicely the idea of a signal cascade, where you have the blue light kicking off kind of like this domino impact of a signal cascade throughout the whole cell, including proton pumping using ATP. And so when you have stomatal opening, you are investing energy if you're that plant. But the upshot of all this is to get a situation where you can get an influx of potassium, which, of course, when you have a big influx of potassium, that makes your psi sub S of your cell more negative, which makes your overall water potential more negative, which makes water rush in from less negative to more negative. And that allows for the guard cells to move away from each other and reveal the pore. So you're not expected to know the details of this, but this is a nice image showing that the blue light is kicking off a signal cascade. And we'll be looking at that in more detail too when we get into hormone impacts, looking at signal cascades. Here is coaction. So this is just, again, I'm not going to get into the details of this. I mainly put this in just to show that you can have these photoreceptors kind of working as a team to help the plant figure out environmental signals and hopefully respond appropriately. So here it's the coaction of phototropin, phytochrome, and cryptochromes. And this is slowing down stem growth in this case. And so it is complicated. It's a lot of very painstaking research to try to tease all this apart. And just like with hormone research, they're realizing that you often have pigments interacting in order to help the plant decipher what's going on around them. And we're going to find out it's the same with hormones. You actually often have hormones interacting in the same manner to help the plant figure it all out. Okay, UV light. We know UV light's not good for you. It's not good for plants either. And so they need a way to try to assess that. For all living organisms, UVB is the most damaging. So they do have receptors. They do have photoreceptors for that. And it's called UVR8, and they have a little mock-up of it there. This one's a little different in the sense that it breaks a dimer bond. Some salt bridges here between the two subunits, and it breaks it fully apart. So that's a little different. And then the broken apart photoreceptor is the one that interacts with proteins in the DNA that are controlling gene expression. So it's actually the tryptophan amino acid. That's what they have deciphered here. The tryptophan that's actually absorbing the ultraviolet B light. So it is, all of these are pretty simple and elegant mechanisms. It's causing a physical shift of the protein or breaking a salt bridge here to get the subunits separated. So pretty simple, but highly effective for the plant to be able to understand that as part of the signal cascade and help it to respond appropriately to that signal. Okay. So that's all I'm going to say about kind of the introduction of signals. But certainly learning about the pigments and the photoreceptors, the proteins, and looking at how they absorb the light energy and how they're involved in helping the plant decipher all these environmental signals coming at them. So I'm going to continue on with this idea. The next slide says posted in EDUCAT. I want to start integrating now the hormone portion of the signal cascade. So if you have a plant that's living its typical life, its roots are buried in the soil. It can't physically get away from that, can't get up and run away. It has to deal with whatever life brings to it. And so when you investigate the plant body and look for, at the DNA level, look for its ability to sense what's going on around it, all these different photoreceptors that they're finding, all the hormones involved, they're realizing that plants are exquisitely fine-tuned to be able to assess their environment and respond appropriately. And some plant physiologists argue they're way better at it than we are, because we have the ability to move. If we don't like a certain environment, we can get away from it. If we're getting attacked, we can defend ourselves to some extent. And so this idea that the plants don't have that luxury really has forced them through the evolutionary process to really have a sophisticated suite of mechanisms in place that allow them to constantly assess what's going on around them, and then hopefully they're going to respond appropriately. So we're going to look a little bit at that. It can get pretty gnarly pretty quickly. So there's certain things I'm just going to kind of keep at the introduction level, and we're not going to go into some of the details. But basically, what we're going to see, whatever hormone we're talking about and what it's doing, you have this signal that the plant needs to pay attention to. And that usually means there's a receptor involved. So the signal will dock into a receptor protein. And then we say that that signal gets transduced. That just means it moves. The signal moves, in this case, typically into the cell, and allows for a shift in gene expression to give a plant response. So that's kind of like the big picture idea. No matter what signal transduction we're talking about, that's kind of the order that they see over and over. You have a signal, has to dock into a receptor, and that's the way the plant gets that signal to be understood. And then that signal has to get transduced into the cell, typically also all the way to the DNA level, so that the plant can respond appropriately. So basically, we're thinking about this idea that you have that external signal, like a sunlight or something, but then you get it converted to another form inside the cell. So you have all these other chemicals that will come into play now and help get that external signal understood by the plant cells, and get it all the way to the shift in gene expression. And so when they studied this idea, they often see that some of these intermediate molecules, we call them second messengers because they're kind of intermediate into the process. They're not the initial and they're not the final. They're intermediate. So they're second to the initial signal. And they often can help amplify that signal that's coming at the plant. So we'll get into that a little bit more. Here are some ideas of plant responses. Often, we point to something that is more dramatic for us to witness, like the Venus flytrap catching a poor fly in its trap. That can be quite quick, and the fly doesn't have a chance because the leaves snap together so quickly. And so it's the action of the fly crawling on the leaves. There's some very sensitive trichomes that are on the inner surface of those modified leaves. And once two trichomes get triggered within a certain period of time, that's the signal to the Venus flytrap. There's something crawling on me. It's not just a twig that landed there with a wind, because it's moving. And so if they feel that movement across their trichomes, that's actually the signal to them. Oh, something's crawling. It's probably something I want to eat. And so they'll close their leaves over the insect. So, by the way, if you ever buy a Venus flytrap, they sell them every once in a while. I see them at like Menards or Lowe's. You don't have to catch insects if you don't want to. You can typically just give them what they need in the soil system, and it's okay. When I had Venus flytrap plants at home, when my boys were little, we deliberately caught flies because, of course, my boys liked that idea. And so we'd hunt for flies and bring them to the Venus flytrap. But when they sell them in stores, they're already well nourished in the soil. So if you go and work with Venus flytraps that are in boggy areas, they don't get enough nitrogen in boggy areas. And so that's why they want to get insects in the exoskeleton. Lots of nitrogen. So it's a beautiful source of nitrogen. So that's why they've adapted that strategy. But if you take them and pot them in a soil with plenty of nitrogen, they don't need flies. So you don't have to go hunt flies if you don't want to. Here's sundew again, another bog plant that is curling around the insect. And that insect is toast, it's going to be not able to get away. And again, so it's that physical sensation. Plants have amazing touch sensors all over their body. So when you touch your houseplant, they feel that. Absolutely, they feel that, and they are responding to that. So you can see touch receptors all over their body. Here in figure C, you can see the wind has consistent wind pattern has really done a number on the morphology of that plant. But it's a way for that plant to adapt. So I'm guessing the wind is coming from the left consistently up over the cliff. It looks like it's on near a cliff here, and is pushing, sweeping across from left to right. Well, if you want to survive that high wind, it kind of makes sense that you bend to let the wind spill off you, so you're not getting such a force from the wind striking your body. So kind of create a shape that allows for the wind to just kind of flow over and off you and not destroy you. What do you think happened with D? Why a tree might do that? There's a tree like this at the island. If you're on one of the trails, it looks pretty much like this. I have a picture of one of my boys when he was a toddler standing on it. My guess is that when it was really young, maybe there was something that fell on it, and so the trunk, went almost horizontal, and then whatever was weighing it down stayed there for a while, and so it started to curve up to try to get back up to sunlight. That's my guess. I know that certain Native American tribes used to do that on purpose. They would bend saplings in a certain way as like a signpost of marking a trail or something like that. So But that's my guess for that. Very odd design to the tree trunk. Here they're just showing, if you cut a tree that's more horizontal, and look at its tree rings, you see that there's some asymmetry here to the tree rings where they beefed up some of their xylem, to again, just give them that extra strength in that portion of their stem. So plant responses can be very slow in the sense that it's a growth pattern shift, like what we see in figure C or D. Definitely a growth pattern shift over time. So you would have to sit there and wait for a long time to see that response. But from the perspective of the plant, it's appropriate. Here in figures A and B, we can sit and see that very quickly with our own eye, just in under a minute. And so these responses can be very quick, where this is more of an well, I won't go into the details. It's a very quick response and is due to touch, whereas these others is still touch, but it's a slower growth response. And so plants are quite amazing at that ability. Some can act very quickly and others takes a while. This is a good flow chart to look at as you're studying, because again, it's giving like the big picture idea of what we're talking about with signal transduction. So you get your environmental signal, the signal transduction, and then what they're saying is it needs to dock into a receptor protein. And then often there's a second messenger involved. So you get amplification. They help. These second messengers are helpers. They help get that signal really firmly established and get it going on the cascade. And then you get the transmission. Often hormones are involved in that, or like with the Venus flytrap, you get electrical signaling that's causing the quick shift in closing the leaves. And then you get some kind of response. So I would like you to know the order of this. This is like standard. You're doing this too in your body. It applies to you too. But certainly plants are also using these types of signal transduction schemes. And then you see this brown arrow kind of going back here from the end more toward the beginning. So what they're saying there is that at some point you got to turn off the response. Like at some point it's like, okay, we've done our job. We don't need to keep responding. And so there has to be a way to kind of quiet everything down. And that's what they mean by attenuation. And usually it's some kind of phosphate shifting going on. So I'll explain a little bit more about that. Or the receptor itself gets degraded down so it can no longer hear the signal, so to speak. So your body's doing this too. But we're gonna, of course, in this class, zoom into what's going on inside plants. So this is a very good general scheme to think about of the whole process. So here's a plant cell. And this is a good slide to study too, because this is getting across the point that you have to have a receptor in part of this process. And so here you're looking at, let's see. Well, they're trying to show that there's different areas in the plant where you can have the receptors. So here's the mechanosensitive channels or receptors. Those are the touch receptors. But they're trying to show that it doesn't always have to be in the cell membrane. Initially, they thought that's where all the receptors would get stuffed, is in the cell membrane. But then they realized that hormone receptors in particular often come into the cell and dock into a receptor inside. They're even showing all the way into the nucleus. So there's different locations. You don't have to memorize, which hormones have which locations. Just understand it doesn't always have to be the cell membrane that we have these receptors. And then when they started looking at the intermediary players in the game and looking at what, besides the signal and receptor, what else is going on, and they see this cascade inside the cell, calcium is actually a huge player, which surprised them, I think, initially, because that was not a big, strong hypothesis when they initially started looking at that. But they have found that calcium, the more they look, the more they find it as a second messenger in the signal transduction. Shifts in pH. That's a pretty simple mechanism, and they see that some plants will do that. They'll have so basically what that's saying is there's some movement of protons to get a drop or a rise in the pH, and that in and of itself can help amplify the signal. They even use some of their own cell bits. So they know that, their cell membrane is composed of lipids, fatty acids and lipids. And they'll actually see that some of those lipids play a role as being a second messenger. So that again was kind of a surprise. And then this group of molecules, reactive oxygen species, or we often just abbreviate that down to ROS, R-O-S, ROS. So you may have learned that, and I've mentioned it, I think, at some point in this class, maybe, that reactive oxygen species, yeah, we have, because when we were looking at damage to the photosystems, we were talking about free radicals and how oxygen can get into a reactive form. So in that regard, it's not good, because it's damaging parts of the photoreceptors that have to do photosynthesis. So obviously not a good thing. However, sometimes reactive oxygen species that can be a very important player in the plant, helping the plant understand what's going on and respond appropriately. So it kind of depends on the context of the situation, the amount of the reactive oxygen species. So sometimes reactive oxygen species are really bad for the plant. It can damage the plant. Other times they're actually using it as a signal. And so that again was another surprise, that is actually can be beneficial. It's kind of weird, but reactive oxygen species can be beneficial under the right dose and context. So this is a very busy slide. And again, I'm not going to go into details here either, but a couple of big picture ideas. So right here, you see the idea of a pH shift. They're showing that there can be protons getting shifted either into or out of the cell. Here they're showing it going out. And of course, that's when you look at pH, it's highly dependent on the hydrogen ion, H plus, concentration. So they're showing that there. All the yellow balls are calcium. So they're trying to show how active calcium is in a variety of ways. All right, so calcium can be a big player, both, well, in many different parts of the cell, actually. And then again, down here, you're seeing using ATP, in this case, to get calcium in or hydrogen in. So you don't need to know the details of this process, but this slide is showing some of what we talked about here, okay, with the second messengers. I don't see them showing any of the ROS idea here, but we'll get into that a little bit too. Here are the idea of membrane lipids. So they'll actually have some of the membrane lipids like break free of the membrane, and they'll be just small bits and pieces of the membrane. And that can be used as part of the signal cascade process. So these are all membrane lipids that they've detected as part of the second messengers. And then this is the idea of reactive oxygen species. So for like right now, if you ate lunch or a good breakfast, your body in your mitochondria is doing this. You have oxygen sitting at the end in your mitochondria of an electron transport chain process. So O2, in order for O2 to be converted to water, it has to accept 4 electrons, but it doesn't do it all at once. It does it one at a time. So your electron transport chain in your mitochondria has to send 4 electrons down that chain. And so it's a stepwise process of accepting electrons. Well, there's a downside to that because as that's happening, you start to get these free radicals in that process. So it's kind of odd. You absolutely need oxygen to survive, to stay alive. But in the end, if nothing else gets you, it's going to be oxygen that gets you. More specifically, the free radicals that form as your mitochondria is kicking along. And so if you don't die from anything else, it's going to be the free radical damage that gets you in the end, because it has to happen in this stepwise fashion. And if you don't have good protection in your body against free radicals, then you have these superoxide anions, peroxide, hydroxyl, all of those can be very damaging. And so that's the same thing with the plant too. The plant has mitochondria and it's doing this. So reactive oxygen species are just a fact of life if you're an aerobic organism like we are and plants. So reactive oxygen species, yes, they can be not so good for you, like what I just described. It can really cause some health issues. But on the other hand, it can help give information. It's part of the signal transduction. And so it is truly a dual role. It has a dual role. Here you're seeing all the things that impact reactive oxygen species in the plant. And so you see a lot of this is abiotic stress. That means environmental stress. And again, if you're a plant, you have to deal with this. You have to be able to tell if the soil's getting too salty, or if you're getting too much light, or if it's getting too cold. You can't just get up and walk away or put a blanket on. All right. And so the reactive oxygen species is part of that understanding by the plant. It helps them understand what's going on around them. I've also typed in, I added this into the figure, biotic also, because we're going to see as we get into plant pathogen interactions that reactive oxygen species also play a very important role. Okay, so it's not just abiotic. But you see that the reactive oxygen species, yes, it decreases. So this red line with the bar, that means it stops it. So yes, if you get reactive oxygen species, it's going to slow down growth and development and yield, especially if it goes on and on and on and on. But you see the green arrow here, that's trying to imply this is a good thing. Okay, green is good. And so they're saying, yes, it also has this dual role of it can impact negatively on growth, but it can help with the signal transduction process so the plant can understand when it's under a certain stress, whether it be abiotic, like all these abiotic things they've listed, but we'll see also with biotic. Okay, so definitely a dual role for the plant. Okay, we'll stop there and we'll continue on. We're going to get into hormones next on Monday and go through the 5 major groups and a couple others.